All right, great. In that case, let's pick up where we left off. So we were building a temporary table called ANOT. And in this table, we're going to build it out with the new gene identifiers that we're mapping onto the existing ensemble identifiers. So our our little table, we're starting it out by just saying, please create us a little data frame with the row names that are the same as the DGE output table. So our DGE output table has row names as ensemble IDs. So we're starting out a table with no data in it, just a row of ensemble IDs, and then we're going to add in columns that map onto those ensemble IDs, but they'll be different gene IDs. So that's what all of these if statements here are for. We're basically accessing the and.dbi variable that we defined, which is when we access that database, and we are getting 5 different types of gene identifiers. So gene symbol, gene name. We're also getting ensemble, RefSeq, and the Entree IDs. So we're querying the database with the existing list of ensemble IDs and saying, bring in all of the gene symbols that map to one of these ensemble IDs. So if you haven't done so already, please make sure to run this cell. You should get a few little warnings here, but this is not concerning. This select returned one through many mapping between keys and columns. This just means sometimes one ensemble ID maps to multiple symbol IDs or gene names or whatever. So note here, each time we run one of these queries, we're adding in a new column using the $ operator. So we're saying in our new temporary data frame, add a new column called symbol and put all the gene symbols there. Add a column named gene name, so on and so forth. All right, so let's use the next code block to look at this little table that we've created and answer a few questions. For these questions, we're going to use polling in WebEx rather than Mentimeter, since I think Lisa and Armin both have meetings. So go ahead and use the head function to look at the first 6 rows of our little gene annotation table, variable name ANOT, and answer these questions. First, how many different types of gene annotations does this table contain? All right, I'm going ahead and opening the poll now. How many different types of gene annotations does the table contain? Going to give a few more seconds. Those of you who haven't had a chance to answer yet. All right, still waiting on answers from quite a few people here. This is not a trick question.
Oh, I answered in the Menti thing first and then saw it in the poll, so maybe some other people did that too. Even though I know you said that out loud, but I didn't register for me.
Ah, okay, sneaky. Let me go over here to, I don't even have, oh, here we go. Oh, yeah, you're right. Thank you, Mike. Several people did answer in the Mentimeter. Okay. So we're not using Mentimeter right now because there's no one, I don't think, online to change the questions, and Amanda and I don't have control over that. That's the GPL folks. So wherever you answered, great job. You get credit. Um, let's go ahead and I'm going to close the poll here on WebEx just for my sanity. And all right, so we can go ahead and go through this together. So head will show us the first 6 rows of the gene annotation table, which is held in the variable anot. And if we take a look here, so I see 1, 2, 3, 4, 5 different types of gene annotations um, in this table. So we started with ensemble, we added 4 new types of gene annotations. All right, and so then let's again do this one together. What is the name, gene name and symbol of the ensemble ID that ends in 28? So we can just go right across here, gene name, cell division cycle 45, or symbol CDC45. So just kind of an example of how we're taking the ensemble IDs and putting them into something that is biologically meaningful and human readable. And then this gene that ends in 31 that we kept looking at, remember the expression was much, much higher, mean overall in the ground control samples. That's H19, which is a long non-coding RNA implicated in brain development, among other things. All right. So before I get into this next section, we are going to be adding in other annotations from the string database. I would like to pause, and we've had an update to our database based on an error that our very own Mike Lee identified. So I was going to give Mike an opportunity to describe what he found in the update that we're going to make to our pipeline.
Yeah, and Amanda mentioned this is a good example. I think Amanda mentioned to me, not everybody. This is a good example to demonstrate how like open source code is powerful because there's a lot of people using it too. One, and not everything is perfect just because it's packaged and available. But the deal here, string identifiers are just another type of ID. They happen to deal with a protein interaction database. And the program here is supposed to take what we give it, our IDs, and use that internally to match them up to the appropriate string IDs. And some things were looking a little funny. And after digging into things, there was a problem in like one of their nested functions, like a function that they have in their package uses a sub-function, uses a sub-function. And I found that there was a tiny little blip in their code that was parsing the IDs wrong. And I'm going to put the link to the forum where I submitted the problem to them. It's pretty dense. You don't have to look at it at all if you're not interested, and I don't think you should read through it now because it'll be distracting. But it just gives you an idea of how like a regular user in the community thinks they saw a problem, and I shared it with them and said, here's what I saw, here's how I think we can fix it. And then and then they implemented the fix, and it's great. I didn't extensively dig into how often it was wrong versus right or anything. And this program does a lot more than just this one. This particular problem was only with the one type of ID we happen to be using. So it's not like their whole program was broken or anything, but it was broken for this ID match. And it's a good example of how open source is flawed, but also really robust because you potentially have all the users around the world to spot a problem and then help fix it. That's why if something exists and is well used, that's generally better to use than writing something ourselves like ad hoc at the time.
Just to follow up with Mike's awesomeness, the developers did get back to him fairly quickly, within like what I think it was less than a week. And then went ahead and took the fix that Mike proposed, implemented it into their code, repackaged it with that fix in it, and then just released a new version of this package like last week or maybe a week and a half ago. And so Mike right now just finished testing it on our end. So we're going to be implementing that changed in the next iteration of our pipeline, which we're going to have come out very soon. So just to note that this version that we're working with does have that issue, but we will be updating it once we incorporate it into our pipeline. So thanks, Mike, for being awesome.
I never get tired of hearing that.
Thank you very much, Mike. Great job. Great example of interacting with open source code. So Amanda, this update you were talking about, is this where we're going to use version equals 12 instead of 11 here?
Yeah, I think it's even more than that's like 12 point some, 12.4 or something.
Nice. Okay.
Yeah, but it you need a different version of R than we currently have because it's installed. It was made for the current version of R and the current version of Bioconductor, so we would have to update a lot of the background packages for this. So just wanted to point that out. It's only going to affect that one column, that string DB, the string ID column. All the other columns are going to be fine.
Excellent. All right. Any questions from anyone about this? Okay, great. Okay, so as Mike mentioned, we are going to be mapping to another type of ID, the string ID. String database contains protein-protein interactions. This is really useful for a functional enrichment analysis. So far we've been mapping to other gene IDs. Here we're actually going to be getting at what do these genes do when they turn into proteins, the ones that are protein coding anyway, and how do they work in protein-protein interaction networks. So we're mapping, we're pulling in any protein-protein interaction networks that contain each gene in our list. So let's go ahead and run this first cell here, which calls the string database and accesses it with the correct, it uses the taxon column from our organism table. So if we go back up to the organism table, there is this column instead of species, we have taxon number. So that's what string needs to access the right organism. And then we also pass in the organism. And we need to use the symbol column from our not table to query the string DB database. So we apparently can't use the ensemble IDs, we use the symbols. And we add in our new string identifiers to the little anot table. So you shouldn't get any output, or sorry, you will get output from this cell, which will say something like, warning, we couldn't map to string 28% of your identifiers, which is fine. A lot of our genes are, nice, we can use ensemble IDs now. Nice, excellent. A lot of our genes are not protein coding, so they're not going to be in the string protein-protein database, so it's fine that not all of our identifiers were found or mapped in string. Let's take a look at our string annotations table that shows our gene symbols mapped to string ID. So this is what a string ID looks like. This identifier within the string database refers to a specific protein-protein interaction network you can look up and see which proteins are involved in pathways and biological processes. All right, so that's string, and then that brings us to the last set of different gene identifiers we're going to add to our annotation table. And that is the GOSLIM IDs. So GO here stands for gene ontology, and each GOSLIM ID refers to a specific gene ontology, which is a list of genes that correspond to a biological function. So gene ontology networks or ontologies can have non-coding genes in them because they include interactions between, like regulatory interactions between microRNAs, long non-coding RNAs, so on. So here we're querying the Panther annotation database. And again, since we set up our organism variable, that's all we need to call the database, let it know we're working with mice, and then we use the entree ID column from our little annotations table to map into the GoSlim IDs. And then we add a new column to our not table called GoSlim IDs. So go ahead and run that column or that cell. So we're accessing the panther.db database. We're mapping from entree IDs into GoSlim IDs, making sure we don't have any NAs. Sometimes when we do this mapping, some of the results have NAs. We don't need that in our table. That's not informative anyway. And then adding in a new column here. All right. Hopefully everyone else's has finished as well, and we can use the next cell, which is pre-populated, to take a look at the first row of our final gene annotations table before we merge it back in with our big DGE output table. So I'm doing head and I'm doing n equals 1. And so please go ahead. This is going to only run the first, show you the first cell, the first row of this table. So please go ahead and run this cell. And then I'm going to put up a poll to answer both of these questions at once. All right, I've opened the poll. So what's the name of the gene that we're looking at? And then how many biological functions involve this gene? And as a hint, please look at the GoSlim ID column. Make sure you press submit on the poll once you're done answering. I see a couple people have their answers still in progress, so I'm going to wait for a moment. All right, in the interest of time, closing the poll now. All right. Nice. Okay, so let's take a look here. So the name of the gene shown, right? Mononucleotide binding protein or G protein alpha inhibiting 3. It's the name, and the gene symbol is GNAI3. And I have been told there are 55 GoSlim IDs here. So lots of different gene networks that involve this gene. All right, so the utility here is we now know a lot more and have a lot more avenues for searching than just having our ensemble IDs. So the last step that we can do here is use cbind to add our ANOT table into our DGE table. And let's go ahead and run this cell, and then we can run the next cell to look at just the first row of our DG output table, look at all the different columns that we've added. And we have to look at only the first row because there are so many GoSlim IDs, it would be difficult to look at more than just one row here. So as you can see, the first the first 6 columns of this data frame now are the different gene IDs or protein, string IDs. Oh, rather, sorry, the first 7 columns here, and that includes the GhostLim IDs, which are gene ontologies. Then we have our sample counts, the statistics from the Wald test, both directions, our mean standard deviation across all samples and within groups. So that's it. We are done making our DGE output table. I've added a list here of all the columns, gene ID systems, protein-protein interactions, functional gene ontologies, so on and so forth. So yeah, right. And so here's a note. This is something we went over earlier. The GeneLab standard pipeline generates the likelihood ratio test p-values, but here we're just doing Wald values. So there are no LRT columns in this output table, so it's not exactly the same as an output table from the GeneLab pipeline. All right. So the last step is to use write.csv function to write out first the contrasts that we used, save those, make sure we can refer back to those later. And we're going to write out our DGE output table to a CSV file called differential_expression.csv. So both of those will go into your DGE output directory in your home dir. All right, I'm going to switch back over to the lecture now and go over data visualizations. But before I do that, any final questions on DSeq2? Okay, great. All right. So I'm going to go over visualizing RNA sequencing data. So I'm going to go over the different types of visualizations and the statistics behind them that we're going to use. And then we will, if we have enough time, we'll come back to the notebook and actually make these visualizations. So please shout out or put something in the chat if you have any questions. So after our DSeq2 differential expression analysis, we may have hundreds or thousands of differentially expressed genes. We haven't looked to see yet how many differentially expressed genes we have. I just know that this may be the case. With this kind of data, it's easier to interpret a visualization or figure. And 2 common visualizations for RNA sequencing are the Heatmap and the Volcano Plot. So we're going to make both of these visualizations using our sequencing data here. We're also going to make a PCA plot using only the differentially expressed genes, but I'm not going to go over PCA because I did that already. All right, so I'm showing here an example of a heat map, and I'm also showing an example gene expression matrix that might lead to a heat map like this. So we have samples as columns and genes as rows with count data in the values. And we can create a heat map like this where each of the range of values is mapped onto a color bar here from -2 to +2. And each little box in the heat map corresponds to that area on the color bar, wherever that number is. So another thing to notice here is that, again, so samples are across the top, and we've performed hierarchical clustering, which shows us which genes are most similar, or which samples are most similar to each other, and same thing along the side for genes. So I have this in a little bit more detail on the following slide. So again, rows are clustered based on the similarity of values. So genes are not in the same, so genes here are in a certain order, gene 1 through gene 18. After running the heat map, the genes are now not ordered numerically, they're ordered based on how similar they are, right? So this group of genes up here at the top right seems to have a specific pattern of expression where it's really high in these samples. This group of genes has higher expression in these samples, lower expression in these samples, and then high again to the left, whereas the other genes down here have lower expression in this group of samples. So that we calculate similarity based on different types. There are different types of similarity calculations based on a correlation or covariance of the vector of values for each gene. And so that's how we get this hierarchical clustering for rows. Same thing for columns. Columns are clustered based on similarity of values, so that's how we get samples that seem like they're kind of all behaving the same way are clustered together. Yes, that's right, legend or color bar, and the colors are mapped to the range of values in the matrix. So smallest value here is -2, and the largest value here is +2. So it goes from dark blue to dark red. So here I've just shown a heat map where the values kind of seem a little bit artificial. They go from -2 to +2, but real data tend not to be that tidy. So these data have been scaled to live only between -2 and +2. So here's a little cartoon illustrating what happens when you scale data. So before scaling, we have these 4 points that look like stars. Before scaling, the points are far away from each other and have larger values. After scaling, the points still have the same relative relationship to each other, but they're kind of all in the same ballpark. They're between 0 and 1, let's say. Scaling in this instance is compression. Compression is one way to think of it. Yeah, scaling is like, this is min-max scaling. So we take the min of the original data, the max of the original data, and map those onto new min and max values. So in this case, the new min is -2 and the new max is +2. And then the rest of the values have to fit on that number bar in between those two values, but at the same relative relationship. So the same relative distance from each other. So pre-scaling, gene count values can range from all the way as high as 30,000 down to 1. And so the larger values can skew the data non-representatively and make it very difficult to look at a visualization. So we'll see an example of that in the notebook. So scaling brings all the values to similar proportions so they can be reasonably compared. But yeah, as Amanda mentioned, when we scale like this, the relative differences are preserved. So we bring them down into similar proportions, but the relationships are the same. So that's why I really like this visualization here, because you can see after scaling, we just kind of have a mini representation of the original data. All the relationships have been preserved. Doesn't this make it harder to call what is a real fold change? Potentially, but we don't calculate fold change on scaled data for that reason. So when we calculated fold change, it was on data that had simply been normalized for read depth. I'm only doing scaling for visualization purposes, not for analysis purposes. So this is just to make it easier to look at relationships in the data, not to say, well, here's a statistically significant relationship. I wouldn't do that after scaling. No worries. Good question. All right. Other questions, discussion points on scaling? All right. So that was heat maps. The second visualization I wanted to go over here are volcano plots. So heat maps, we were just looking at count data and mapping it onto a color range and looking at those values. Yes, after scaling, but we didn't do any statistics like log full change, p-value, etc. Volcano plot is a way specifically to look at the statistics your in your data through a visualization. So here, all right, so I'm showing here on the left the results of a differential expression analysis. Maybe we have some gene names as row names. We have our log2 fold change that we calculated. We have our p-value and our adjusted p-value columns. So in a volcano plot, we're able to visualize each dot as a different gene, and we can break down our data set by genes that did not pass our adjusted p-value cutoff, whatever we set that as. It's also traditional to set a log two-fold change cutoff because you can have genes that are significantly differentially expressed, but since we noted that at very low expression levels, RNA sequencing data is noisy. So it's important to also have a log two-fold change. You want to make sure that a log two-fold change cutoff to make sure that you're not just picking up on noise in your adjusted p-value. You actually do have a large difference in the values of expression between your two groups. So we set a log two-fold change cutoff. People set this at 1 or at 2. There are a few different accepted values here. So in a volcano plot, we can see, all right, here are a bunch of genes that didn't pass either our adjusted p-value cutoff or our log two-fold change cutoff. We have a bunch of genes that only, in blue here, only passed our adjusted p-value cutoff. And I'm going to break down this plot into the different parts in a minute. I just want to point them all out first. So blue genes that only passed our adjusted p-value cutoff, and green genes that only passed our log two-fold change cutoff, and then red genes that passed both cutoffs. All right, so breaking this down a little bit, if our log two-fold change is in yellow here, and our adjusted p-value is in green, note that the y-axis is a negative log 10 of the adjusted p-value. That way, the axis is just very intuitive. So, right, as adjusted p-value gets smaller and smaller and smaller, it gets more and more important. That's a little bit difficult to visualize on a continuous axis. So instead, we take the negative log 10 so that everything is inverted and we're now on a constant scale. And we can say that as a negative log 10 adjusted p-value increases in value, the more significant that result is. So higher is better for our y-axis, so negative log 10 adjusted p-value. Yes, so in RNA sequencing data, we use adjusted p-value as a standard rule. And so if I'm going to make a volcano plot from RNA sequencing data, and I'm going to say, here's a bunch of genes that I think are significant based on p-value, I'm going to use adjusted p-value because we have to correct for multiple testing comparison. Is that in RNA-seq or in proteomics, Joel? Do you perform multiple testing correction for proteomics the same way we do for RNA-seq?
Yeah, we do all that. We do it. We still just present the just a raw volcano plot, because otherwise you have the compression and it doesn't actually look like a volcano. It looks like a compressed little winged graph.
Yeah. Yeah, that's true. Yeah, I just want to only see the genes that I think are significant based on passing the adjusted p-value cutoff.
Yeah,
Uh,
we do that. We offer the option to do that, and we will do
okay.
that when we're personally looking at it. But generally, most publications, we just present it as is, and sometimes we will color them as well.
Yeah. Okay, interesting. Is this
Yeah, it's my
your core?
lab's. Yeah, our core. We were the first ones to publish a Shiny app on analyzing proteomics data. There's a demo there if you wanted to see what it was like in your own
Th
personal time.
Thank you. I do. Yeah, appreciate that. I'm always interested in the how to analyze proteomics data because I've generally just analyzed RNA sequencing, but I have dabbled in proteomics. So maybe we can chat offline. All right. So I was mentioning that we use the negative log of the adjusted p-value um, because then as the adjusted p-value gets smaller, the negative log adjusted p-value gets bigger, and it makes a nice intuitive plot. So we have significance cutoffs. Here on this plot, we've set significance cutoffs of, I think, like 1 and 5 for the log fold change and the negative log adjusted p-value. And so here, just to break down this plot, a gene is colored green if its log fold change between the compared conditions is greater than the absolute value of 2. So that is covered by these two areas on the left and right hand side of the plot. So +2, -2. And then a gene is colored blue if its adjusted p-value before we take the log is less than 10 to the -6. So that is everything above the horizontal line. And then a gene is colored red if its log fold change is greater than absolute value of 2 and its adjusted p-value is less than 10 to the -6. So in the volcano plot, the point is we can kind of stratify the results of our differential gene expression analysis on whether a gene passed log two-fold change cutoff, adjusted p-value cutoff, or both. So that's how we get a plot that looks like this, and we have labeled some of these genes here. So red is past both cutoffs, blue is past only the adjusted p-value cutoff. So that's anything above the horizontal line, but not on the outside of the two vertical lines. And then green is past only the log two-fold change cutoff. So that's on the outside of the two vertical lines, but below the horizontal line. Right? Any questions about volcano plots before we get into making some of our own? All right, great. All right, so let's go back to SMCE Jupyter Hub. So back at SMCE Jupyter Hub, the last section in this notebook is creating 3 visualizations using our differentially expressed gene data. So everyone please double check that the kernels are still live. You're running the right kernel, and it should say idle because we're not running anything right now. Is anyone having any issues with the kernel? Okay, great. So the first thing that we're going to do is perform a log transformation of the normalized expression data. So again, we're adding our pseudo count. We have our norm counts, which is the variable holding our normalized expression data. And we're assigning this to a variable called exp_norm. We do this because it is, this is another way of transforming the data to make it easy to look at in a visualization. So now we have our entire DG output table, which has the 21,000 genes, and it has adjusted p-value and log2 fold change cut information, but no cutoffs have yet been implemented. So what we want to do is figure out which genes from the DSeq2 wall test, which genes are actually significantly differentially expressed. So we want to filter to just the genes with an adjusted p-value less than 0.05 and absolute value of log2 fold change greater than 1. So these, as I mentioned, there are a few different cutoffs that are acceptable in bioinformatics, and these are accepted for, this is a conservative way to identify differentially expressed genes. All right, so now what we're going to do is use this next code block to actually get the list of differentially expressed genes according to these cutoffs. So here I'm filtering the DG output table. I'm taking out the adjusted p-value column where we compared flight versus ground control, and I'm saying give me everything where that column is less than 0.05. So that's filtering by adjusted p-value. And then I have two lines where I take a look at the log2 fold change column. I say give me everything greater than 1 and everything less than 1. I merge all that together and remove any rows that might have an NA. So let's run that cell, and we can take a look at the new filtered DGE table using the dim function. All right, so let's go ahead and answer the following questions. We just discussed adjusted p-value, why the adjusted p-value was used to determine significance rather than p-value. That's because for RNA sequencing data, each gene is treated as a different hypothesis, so we have to perform multiple testing correction. Now, how many significant differentially expressed genes did we find? I'm going to go ahead and put a poll up. Although I do see, Lisa, are you back? Oh, perfect. Okay, so sorry, no poll. We're going to use Mentimeter again. Thank you for keeping up with us. So the question is, how many significant differentially expressed genes are
Hey, Lauren, hate
there?
to bother, but can you re-explain why adjusted p-values used to determine significance rather than p-value again?
Oh yeah, absolutely. No bother. Thank you very much for shouting out. So let's go back here. So, okay, right. Okay, so every time when we do differential gene expression analysis, we're testing a hypothesis for each gene. And the hypothesis is this gene is differentially expressed between two groups. We are trying to disprove the null hypothesis, which is this gene is not differentially expressed. Well, when we do this, like as a data set, we're doing hypothesis testing and we're testing thousands of hypotheses at one time, which means that just by chance, we expect there to be a lot of false positives. So if we're using the 0.05 p-value cutoff, that's a 1 out of 20. So just by chance, if we're testing 20 genes at a time, we would expect one of them to have a false positive result just by chance. So in order to take into account the fact that as we're doing thousands of hypotheses at a time, we expect a certain number of them to be false positives, we do a mathematical adjustment of our p-values to take into account that a lot of them are probably false positives. So we basically make it harder to achieve this cutoff by doing a mathematical adjustment that in general makes all of our p-values larger, and then calls them adjusted p-values. So you have to be really, really significant to not when we do the adjusted p-value adjustment, when we do the adjusted p-value calculation, we're basically increasing the value of all those p-values. So you have to be really a very, very small, very significant p-value to not then, pass this cutoff and not be significant anymore after the adjustment. Does that make sense?
Yes, it does. That actually cleared it up a lot. Thank you
Okay,
very much.
wonderful. Yeah. Do you have to do a tuning step to make sure you aren't losing too many true positives? This is a difficulty. So some people would argue that, for example, Bonferroni multiple testing correction, which I believe is what DESeq2 uses and a lot of methods use, is too stringent. So we kind of know we are losing some true positives. But the thing is, without any ground truth, it's difficult to do that tuning step. Like if you are doing an experiment you haven't done before, you don't know which genes are supposed to be true positives and true negatives. So the best that I can generally do is to do accepted best practices, like Bonferroni multiple testing correction, and say, I know what I am seeing will be true positives. I may have lost some true positives, but at least I know I'm not looking at false positives. There are situations where you can set up an experiment where you know what ground truth is, and so you know how, like, you can kind of calibrate for that, but you have to set up your experiment in advance that way. All right, so with that in mind, having done our adjusted p-value cutoff and our log to full change cutoff, we filtered down to a certain number of differentially expressed genes. So returning to Mentimeter, how many significantly differentially expressed genes did we actually end up with? Great, so let's see what everybody said here. So I see a lot of the same numbers, 773. And that is correct. So if we look at the dimensions of this matrix, where I filtered down just to the differentially expressed genes, we know that DIM in R reports the dimensions in rows by columns. And so we know that our genes are in rows here, so that's 773. And then 33 is the number of columns in our DG output table, because that's where we were doing the filtering. So we have 773 differentially expressed genes between flight and ground control. That's pretty good. It's a lot of genes, even using the adjusted p-value. All right, and so just to really hammer this point home, based on the adjusted p-value cutoff used, so that's 0.05, how confident are we in percent that these genes are actually differentially expressed? So go ahead and put your answer in Mentimeter. So 95%, very confident, yep. 99, 95, 95, 95. Okay, so yes, very good. Recall that when we are using a cutoff of 0.05, the p-value is the probability that our null hypothesis is true. So if we use a p-value cutoff of less than 0.05, what we're saying is we have only 5% or less confidence that the null hypothesis is true. So here our null hypothesis was how is each gene differentially expressed? So if we only allow in genes where the p-value or here adjusted p-value is less than 0.05, what we're saying is we are only 5% or less confident that the null hypothesis is true, which means we are 95% confident that our alternate hypothesis is true, which is, yes, this gene is differentially expressed. Does that make sense to everyone? Okay, great. Thank you for the thumbs up. All right. So now, finally, we've been working with the DGE table, which has all those extra columns. We just need to get down to just our counts data so that we can make some visualizations. So first, we're going to create a subset expression matrix containing only significant differentially expressed genes. So that's just, we're just subsetting our x underscore norm variable data frame up here, which has our norm counts that have been log normalized, log transformed rather, and we're subsetting that using the ensemble gene IDs from our filtered DGE table. All right, so if we take a look at the dimensions here, we have 12 samples, which is what we should have, 6 flight, 6 ground control, and 773 differentially expressed genes. All right, so we're going to perform a third principal component analysis using only the significant differentially expressed genes. And this is just something that I like to do. We looked at the entire data set, all the genes, and now we've just subset only the genes we know are differentially expressed between flight and ground control. And I just like to see how that plays out in the global patterns of the data set in a PCA plot. So once more, let's calculate our PCA and hold it in a variable called PCA underscore DGE. And let's create a PCA plot and save it. So comparing this PCA plot with the ones that we've made before, let's think about a couple of questions. So how has the percent of variance explained by PC1 changed since the last PCA plot we made, which was with the normalized data, but with all the genes? So go ahead and put what you think about this in the Mentimeter. All right, I'm going to go ahead and stop voting now. So looks like there's quite a bit of agreement here. So everyone is saying that the percent of variance explained by PC1 has increased, and several explanations, generally getting at the same thing, filtering down to just the genes that are differentially expressed between flight and ground control. So if you take a look here, right, so PC1 is now explaining 56% of the variance. Remember previously when we made the PCA plot using all genes, after normalizing, the PC1 only explained 20% of the variance. So it's gone up significantly. And remember that PC1 has always been the axis that shows the difference between the two biological groups, which are the two groups that we used to perform the differential expression analysis. So we literally selected for genes that would cause PC1 to explain more and more of the variance, because now we're only looking at the relationship between these samples in the context of the genes that make these samples the most different. So this is just kind of a sanity check that our differential gene expression analysis is delivering us genes that make sense. Yep, we see big difference between flight and ground control when we just look at the relationship between these samples from just those genes. All right, what about PC2? Is there anything interesting to note about PC2 or how the relationship between samples changed within the plot along PC2? Let's go ahead and close voting here. So a slight increase in the percent of variance accounted for by PC2. For flight, variance increased. For ground control became better. Flight group is now broken into 2 distinct clusters. 2 points of flight, 1 bioweb likely is a clear outlier. Percent variance explained by PC2 and up. Did not change dramatically overall, but achieved more pronounced cluster separation. Did someone drop 2 flight samples? Yeah. Only increased slightly, flight middle, 2 samples. Yeah, so you're all kind of getting at the same thing here, which is great. So our original, after normalizing the original PCA plot, PC2 accounted for 11% of variance, so it's gone up slightly to 13%. Not nearly as drastic as PC1. And what some of you noted was, if you look at the separation of samples along PC2, there is a big separation between two of the flight samples and the other 4, whereas the ground control samples became really, really close together. So when we subset just to differentially expressed genes, it seems like what we've done is not only have we selected for genes that separate the flight and ground control groups, but we've also somehow exacerbated the technical differences within the flight group. So we will investigate this more tomorrow when we make the heat map, because these patterns are going to become more clear. So that brings us to the end of the day today. Thank you all so much again for your time and your engagement. Please make sure to save this notebook. Click the floppy disk icon, and you can download our progress for the day by going to File, Save and Export Notebook As, and HTML to save it locally. Please make sure that you kill your instance by going to Hub Control Panel and clicking Stop My Server or logging out completely. We'll see you all tomorrow morning.